Everyone, it's Mr. Sinti, and today I'd like to talk to you about the inducible operon, known as the lactose operon, and it's one of the great discoveries of those two French scientists in 1961, and it's a classic in biology because it's a discussion of gene regulation, in other words, transcriptional control in prokaryotes. And we really were fascinated by this whole idea. And let me sort of just jump into it and get into this conversation with you. So you can see in front of us, we have a glass of milk. The reason I have this glass of milk in front of us is that you may be familiar with this already, in that milk has a special sugar in it called lactose, and it's a disaccharide. And milk, you might also know, comes from the mammary gland, and so female mammals produce milk in order to provide nutrition for their babies. And so this disaccharide needs to be broken down and digested into two monosaccharides. And so what's interesting about this is that bacteria, for example E. coli, they live in our intestines. They live in our large intestine, in the colon. And what's interesting about that is that milk is, if you will, a sort of a rare substrate. How often does it come? Well, it comes every time an animal drinks milk. And so as it turns out, E. coli, if you're E. coli's perspective for a second, you're sitting in the colon and you're waiting for milk. It's like, when is she going to drink milk? It's like, well, maybe she'll never drink milk. And if lactose is not something that's present very readily, it'd be very wasteful to produce the enzymes necessary to digest it. And so maybe a better approach to something rare like lactose would be, for example, since you can't control the environment, it's totally unpredictable, maybe it should be a situation where you turn the genes, you keep the genes off, or they're in an off position. And when they're in an off position, when lactose comes your way, then you can turn them on. And so that's exactly what happens in E. coli. Lactose itself induces the production of the very enzyme necessary to digest it. I find that to be quite remarkable. And so just to refresh you on the structure of a disaccharide, this happens to be an example of a disaccharide right here, lactose. It's made up of this monosaccharide galactose and this one glucose. You might be familiar with this bond. This is in a beta orientation, meaning that it's in an upward angle, and it's referred to as a beta 1,4 galactoside linkage, or just a sugar linkage between two monosaccharides. Nevertheless, it's difficult for hydrolysis to take place. Water has a hard time cleaving that bond unless there's an enzyme that's going to catalyze it. So most people on the street might know that the enzyme that breaks down lactose or hydrolyzes lactose is called lactase, but the official name of it is beta, in reference to the bond, beta 1,4 galactosidase. That's the real name of the enzyme. So that enzyme is produced, it's cleaved, and it's digested into two monosaccharides. So that's the background. If you're growing bacteria, let's talk about bacteria for a second. If you're growing bacteria in a test tube, or if you're growing it in a larger glass or flask, you can put a growth medium into it, like something like LB, Laurier-Bertini broth, and at time 0 you can inoculate that with a little bit of starter bacteria. And over a period of time, the E. coli divide and the numbers increase and numbers increase and increase until it becomes very cloudy. How do you know that it's becoming cloudy? You could look with your eye and notice how something that was once kind of a clear yellow has turned into a cloudy yellow. You can also use your nose and smell the fact that bacteria is growing. You can be more quantitative about it in terms of, like, you could take, like, you might recall this from the lab, you can take out small samples from the growing flask and actually place it in the spectrophotometer and shoot light through it, and you can actually measure the the percent of transmittance. So something that's becoming more cloudy over time is less transmittance, and so you can see a familiar growth curve from this. So I just want to refresh you on that. And so here's a piece of graph paper. Like for example, if I had number of cells over here, or just simply n, the number of population over time, and probably a good time would be hours in E. coli because they divide very rapidly. You might recall that at first the bacteria grow rather slowly, and it isn't so much that they grow slowly or they divide more slowly. It's the fact that there's so few numbers, and so the population grows and doubles, so it is exponential growth, but there's that lag phase. You remember this, where when there's a few individuals in the population, they're growing exponentially, but when there's many more, you get a very rapid growth, and then eventually they'll run out of media, they'll slow down, and then there'll be zero growth. And so that's our classic logistic growth curve. So now that we're familiar with that, so check out what happens. This is an interesting kind of experiment. Let's set up the same thing. We'll go number of cells over here, and then down here we'll do time. And this time in our media, in our flask like this that we're growing the bacteria in, let's give it some nice yellow right here, some media. You're like, well, what's in the medium? Well, basically we can control it. We can put anything we want in it. What would you say if we were growing E. coli and we were growing it in a medium that contains, and let me just emphasize this, that contains both glucose, which is a monosaccharide, and the sugar lactose? What's going to happen now? So here's our LB, and then we add some glucose, and we add some lactose. I'll put a G there, some L there, and then we, of course, add the bacteria. And so they're growing. So what do we see? Let's see. So at first, they're lagging like this, and then they're growing exponentially like this, and then they start to slow down. They reach sort of this plateau, and you're like, gee, nothing new is happening. And then they start to grow exponentially again, not as fast, and then they reach another plateau up here. And you're like, okay, this is something that maybe we didn't predict was going to occur. And you're like, how did we even get these numbers? Like, what's going on with that? Again, you can take samples out and put it in the spectrophotometer, and you can come up with this. So what's interesting about this, in certain time intervals, as you look at this, you might ask yourself the question, I don't know, what's the E. coli eating? Is it eating both lactose and glucose in the beginning? It seems like they would. You're giving them two sugars. Why not eat both? As it turns out, when you look at the test tube, when you're at each time interval, it's clear that the amount of lactose in the test tube over time stays the same. So in other words, they're not touching the lactose. They're not digesting the lactose in the beginning. They're eating only glucose. And you're like, well, you don't know that. Yeah, I do know that, because as it turns out, you can test the amount of glucose in the test tube over time, and the amount of glucose is decreasing. So check this out. The reason that they slow down is that they're running out of glucose. There's no glucose present at all at this point, but there's plenty, so that they're eating glucose, but they're not even touching their lactose. That's weird. And then they start to digest the lactose at this point, and that's what gives them the energy that they need to continue to grow until they run out of both glucose and galactose, which is up here. You're like, wow, this is a little weird. You know, it's like, well, what about that enzyme that we were looking at just a second ago, lactase, or beta-galactosidase, if you will? It's like, well, was there any of that enzyme present? In other words, here's the question. Let me use a different color. Here's the question. Is there lactase or beta-galactosidase present right here when they're eating their glucose? And the answer to that is no. There's no beta-galactosidase present. But you may have predicted it. There is beta-galactosidase present here. So, in other words, lactase. So how do you like this? How do you like the fact that E. coli is only eating glucose at first, not even touching its lactose, and then when it runs out of glucose, no glucose present. Let me write it in. No glucose present, but yes, lactose present. It starts to grow again, and so lactose seems to, if you will, induce the production of the enzyme necessary for its own digestion, lactase. This is incredible. This is very incredible. This means that the simplest organism on the earth, a prokaryote, E. coli, is able to regulate its genes. In other words, it's able to control transcription. It's able to keep the lactose operon off, and then it turns it on. So let me repeat that. It's able to turn the lactose, or it is off. The lactose operon is off, and then it's able to turn it on. And you know, it's interesting. You're like, well, this is simple. Lactose induces the production of lactase. But what's even more remarkable is that it specifically chooses, this is cool, it specifically chooses not to go for the lactose while glucose is present. It seems like it prefers glucose. That's a good question. Easier, it doesn't need to be digested, it's a good source of power, energy, it's metabolized very quickly. It's fascinating. And so this is our conversation that we're going to have. So at first, I mentioned this, at first they, I'll go green. At first they eat glucose like this, and then they run out, and then they stop growing, and then in the second growth period, they're eating only the lactose. And it's like, well, they're not eating glucose because they finished it. So how about if you measured the amount of glucose and lactose at each time point? So the amount of glucose would be disappearing, lactose would stay the same, and up here lactose would disappear to eventually. So the big question is, here it is, the big question is, how does the media, in other words, the environment, how does the environment induce the production of lactase? You might suspect that lactose has something to do with it and glucose has something to do with it. And if that's your hypothesis, I think you're onto something. But if you wanted to pause the video now and just sort of ponder this for a little bit, you're welcome to do so, or because I'm going to continue with this. So here's our two French scientists, and this is Jacob and Manon, if I'm pronouncing this correctly, 1961. So here's the big take-home message, the THM. Lactase, beta-galactosidase, is induced. So that means it's facilitated to be produced. It's caused to be produced in the absence of glucose and in the presence of lactose. How do we know that? What do you mean, how do we know that? That's what the data, that's what we just saw in that graph. We know that. So when they're growing bacteria, that's what so here's the question. Why do the bacteria do this? How is it that they're induced by lactose when there's no glucose around? So this is the question. And then, why do bacteria wait? Why not use both? That's a fair enough question. So there's a lot of questions. This is a Nobel Prize winning experiment, by the way. And so this is what we have. Like the Tryptophan Operon, and if you don't mind, I'm going to just review that for a second. The Tryp Operon is a collection of genes, as you recall, in E. coli that all correspond to the production of the biosynthetic pathway that produces the amino acid tryptophan. And we said that it was a repressible operon, meaning that when tryptophan is in high concentration, it binds to a repressor protein that's produced all the time that's inactive, which is weird. It activates it, and then it attaches to an operator sequence in the promoter which prevents transcription. It makes perfectly good sense because if you're given free tryptophan, why would you make it so? The tryptophan itself represses transcription. But this is like the opposite of it. In contrast, the lactose operon is inducible. In other words, it's mostly off. You don't want to produce lactase until it comes. So you want to be able to turn it on. So it's referred to as inducible. If I induce you to do something, that means I'm getting you to do something. It's stimulated when something attaches to it. So you might be able to predict this. In the lactose operon, the regulatory gene produces an active repressor protein, and it bonds. Exactly. So take a look. Here's the regulatory gene. Sometimes it's called Lac I. The regulatory gene is always being transcribed, and it's producing a repressor protein. Now, this repressor protein is active. I just want to emphasize that, active. I'm going to highlight that. So this repressor protein right there is active, meaning it's good, which means that it's going to come up here, and the repressor protein is going to bind right in there. And so you know what this means? You put a tree on the track, and RNA polymerase is not going to be able to transcribe the structural genes. What are the structural genes? Well, one of the structural genes I mentioned before, the main ingredient, is lactase, beta-galactosidase. The second one's kind of cool too. The second enzyme is a permease. If I can over here for a second. Here's E. coli. You need a transport protein, a lactose permease, even to get lactose into the cell. I find that kind of cool. So the sugar lactose actually induces the production of the enzyme that digests it and the protein that transports it into the cell. Although, if you're following that completely, you might be curious, like if this is always in an off, and that's what I'm saying. It's in an off position. This thing's blocking. You might be wondering, well, what's the story? Well, the story is the repressor protein doesn't work perfectly. It isn't on there constantly. Sometimes it's on, sometimes it's off, but it's mainly on. It's mainly on. Therefore, the genes are turned off, but occasionally there's a little permease production. But as it turns out, once lactose gets into the cell, lactose, let me emphasize it with red, lactose itself, the disaccharide, attaches to a protein. This is so interesting. A disaccharide attaches to a protein, changes its conformation, causing it to fall off the operator sequence. And when it falls off the operator sequence, RNA polymerase is able to cruise along and transcribe the genes necessary to digest it. And then when the lactose is all chop, 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 no more lactose, then there's nothing binding to the repressor protein, inactivating it, so therefore it goes back there and gets hooked up. And so check this out. Here's a little video animation showing what I was talking about just a second ago. Let's go here. Let's say that this is the repressor protein and it's active. In other words, the regulator gene is producing this, and where does it bind? It binds to the operator sequence. But here comes lactose. So when lactose attaches to it, it becomes inactive and it falls off of the operator sequence. And when it falls off of the operator sequence, then RNA polymerase is able to bind. I find that to be extremely interesting. So let's go back up here, if I can push the right buttons perhaps. And so another cartoon drawing of this. Repressor protein, it always binds. Look, there it is binding to the operator sequence. And then lo and behold, somebody drinks milk, and oh no. And so milk goes down into the colon, and the E. coli is like, whoa, 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 whoa. There's lactose present. So lactose then attaches to the repressor protein, making it inactive. So in other words, turns the genes on. So it induces. Why does it induce? Because now RNA polymerase attaches to the promoter, and it cruises along and it makes the enzymes necessary. That third gene, don't worry about it. No one knows what that does. At least I don't. And so the Lac Operon is a series of genes that codes for the enzymes that play a major role in the hydrolysis of lactose, which is beta-galactosidase. And so the repressor protein's always on, blocking transcription. So in other words, if you go into a room, the lights are off. The lights are off. Or if no one's in the room, the lights are off. And then when someone comes into the room, the lights turn on. So when lactose is present, it induces the production of lactase. And so, little details at this point. So the sugar lactose is actually not specifically the sugar that attaches to the repressor protein inactivating it. As it turns out, it's an isomer of lactose. It's called allolactose. It's a little bit of a detail, that's fine, but it's basically lactose. We also discovered there's a chemical called Iptg. It's also an inducer. It's not lactose, but you can, and I bring this up because we're going to be working with it in the lab, you can add this to the media when you're growing bacteria, and it sort of simulates the presence of the sugar. And so when E. coli, when E. coli are grown with, with Iptg, it acts as an inducer and it causes beta-galactosidase to be produced, and so that's important. So as you can see here, here's RNA polymerase. Apparently the repressor protein's been inactivated, and so there it is producing the messenger RNA, goes to the ribosome, and here it is making the important proteins. So let's sort of talk about this. When beta-galactosidase chops up all the lactose, there's going to be no more lactose, so what happens now? Then the repressor protein comes back up and stops it. So that's very cool. So in conclusion, I want to add one more level of detail to this, if you don't mind, because we never really answered the question completely. I answered so far, I talked to you about how lactose, so if you remember the E. coli growing, I talked to you about how lactose can induce the production of beta-galactosidase. That was that second half of the curve. But why in the beginning of the growth curve, when glucose was present, we didn't have any production of beta-galactosidase? What was the story there? As it turns out, we've now learned that there's two regulator proteins that can control transcription of the lactose operon, two of them. One of them I just mentioned a moment ago, which is the repressor protein. Remember how the repressor proteins always produce active, and then the inducer, allolactose, makes it inactive? So that's regulatory, meaning that lactose can induce the production. In other words, the repressor protein can stop transcription from occurring. Stop transcription. So that means that when lactose is present, there's going to be an induction of lactase. So that's what we talked about so far. So let's talk a little bit about glucose. There's a second protein involved. And this second protein is called catabolite activator protein. The acronym for it is CAP. So it's a CAP protein. There it is. It's a protein. So as it turns out, this protein is produced, and it's produced in an inactive form. And when a chemical, don't freak out, but when there's a chemical called cyclic AMP attaches to the active site of CAP, it makes it active. And active CAP attaches to its own site on the promoter. It kind of acts as a sigma factor. In other words, without an activated CAP, RNA polymerase can't really bind to the promoter efficiently. So RNA polymerase needs CAP. And so as it turns out, the thing is, it only binds if cyclic AMP is attached to it. And you're like, okay, where's this going? Well, this is a picture of cyclic AMP. AMP is adenosine, there it is, and adenosine monophosphate. So there's the sugar ribose. Here's AMP. We're familiar with this. AMP, ADP, ATP, but it's called cyclic because it actually hooks around like this and connects to the carbon. It's kind of weird. So this cyclic AMP, and then let's note this. This is rather important. Let me emphasize it. So when glucose is present, so that means like early on in that growth experiment, the concentration of cyclic AMP is very low. And if it's low, that means it's not going to bind to the CAP, which means the CAP isn't going to bind to the promoter, which means RNA polymerase is not going to efficiently transcribe the genes for lactase. You're like, so what does it all mean? Well, what it all means is that when cyclic AMP is present, RNA polymerase can bind and therefore transcribe the gene. So you've got basically two proteins that are playing with this lactose operon. You have a repressor protein and you have a cap protein. Both of those are important in the regulation of the enzyme lactase. So I hope that was clear. It's kind of a complicated mechanism, and I've got one more thing to maybe, if I wasn't as clear as possible, perhaps this might be the clincher for you if you were to see that. Let me come over here. Let's see if this is helpful. So if I
in Escherichia coli, two regulatory sites, the cap binding site and the operator site, function together to ensure that the three proteins required for growth on lactose are produced only when glucose, a more efficient substrate, is absent and lactose is present. The cap site is a region of DNA adjacent to the promoter. In the absence of glucose, a high level of cyclic AMP, CAMP, builds up in the cell. The CAMP binds to the catabolite activator protein, CAP, and activates it. The cap, with bound CAMP, can now bind to the cap site and bend the DNA, allowing RNA polymerase to associate with the promoter. When glucose is present, CAMP levels are low, and the CAMP does not bind to the cap. The unactivated cap is unable to bind to the DNA, and RNA polymerase does not associate with the Lac promoter.
Kind of sad.
There is a second regulatory protein called the Lac repressor. This protein is capable of binding to the operator site and preventing transcription when lactose is absent and the three proteins of the Lac Operon are not needed.
So, in other words, that was in the growth media in the very beginning.
The repressor covers part of the promoter when it binds to the operator, preventing RNA polymerase from making messenger RNA. When lactose is present, a lactose isomer binds to the repressor and inactivates it. This prevents the repressor from binding to the operator site. RNA polymerase can then associate with the promoter and carry out transcription. Transcription of the Lac genes is said to be induced by lactose.
There you have it, the Lac Operon and Inducible Operon. Thanks for watching.